biggest positive of Brexit was that people now talking about issues as opposed to arguing with each other. Next,
no, we are still.
I'm sorry. I was just trying to go from one place to another. We can continue later. So our next speaker is Schroeder, and it's very close. Treating animals, humans, and cancer and coughs.
So actually, it's not about the ethical side of it. It's a bit different. Side. Absolutely. And so last year I gave a talk about our main, our primary work in the lab, which is in the field of pharmaceuticals. And this year I didn't want to bore the audience. Some of the people were here last year too. And I did want to talk about another aspect where nanomedicine actually has been taking us over the past several years. And it took me surely by surprise, but it is a new exciting world of application. From the world of human medicine to the world of animal medicine, and also today crops. So this is human targeting. I won't be talking about it, but I will be mentioning how we reached animal targeting using these systems. And our primary focus in the lab is really treating metastatic cancer and using different systems for treating metastatic cancer. And the systems we use are used for delivering medicines are usually lipid-based drug delivery systems. We have to modify them for the different cargoes, different payloads, and today also for the different systems. Different organisms actually respond differently. If we're treating shrimp or fish in the ocean, the particles are actually different in their stability than the particles we'll use for treating humans. There's another issue, which is the cost issue, when we want to take nanomedicine from one area to a new area. And where it all started is in RNAi, or inhibiting protein synthesis using nanomedicine. At the time, we were working on RNAi and trying to inhibit genes in a cancer model. However, the history of RNAi began in agriculture. The petunia flower that was described by Napoli and co-workers, where they tried to encode for a stronger, more beautiful color. They introduced a gene and the color was all disappeared. They couldn't really explain it, and Fire and Mello in '98 actually gave the explanation, which they won the Nobel Prize in 2006 for, explaining how introducing RNA into a cell can actually stop the production of a protein inside the body, or also inside of plants. So when we look at the major scheme, it's DNA encodes to RNA encodes to protein, and what RNAi actually explained or discovered is that small RNA molecules can also inhibit the production of proteins inside the body. And that's where it started. But when we look at delivery of RNAi, or of the delivery of RNA in order to introduce RNAi, there's a major challenge. We're talking about negatively charged polymers that need to enter the cells of the body. And I think it was put most beautifully by Nobel laureate Phil Sharp, saying that the three biggest problems we have with RNAi therapeutics remain delivery, delivery, and delivery. How do we take a negatively charged polymer and insert it into a cell? We know that many, many years of evolution actually stopped the ability of RNA or of viral RNA to enter our cells. So what we were working on is the delivery of RNA. And at the time, we produced a new set of molecules. They were based on polyethylenimine, which is a positively charged polymer. It's been used in the past for gene delivery. We used a shorter form of this of polyethylenimine because it was less toxic to the body. In addition, coming from the world of lipids, I knew that the body recognizes lipids very well, and I wanted to disguise the polymer, the cationic polymer, with lipid tails. So using an epoxide ring addition, we connected different lipid tails to the cationic backbone to get a new set of polyethylenimine with lipid tails. So we could use PEI with different lengths, and we could charge it with a number of tails, or use tails of different lengths. And we screened all these compounds as RNA carriers in the beginning for HeLa cells and then for other cells. So a hit would be a material that was able to carry the RNA into a cell and induce knockdown that was below 20%. That's where below 80%, and meaning that we'd have still maybe 10 or 15% expression. We went on and tested it in 2 additional cell lines in order to reach the in vivo trials. And we went on to the in vivo. Oh, we went on, these are the 2 additional cell lines. And then again, we screened out materials that were ineffective of delivering RNA to the 2 additional cell lines. And now we moved on to the in vivo. But I would want to give one word about the particles and how one particle for RNA delivery will be successful and another was not successful. So all the particles we created were around 50 to 70 nanometers in diameter. They form spontaneously. And you can notice in a cryo-EM image right here of the particles where the darker region between the lipid bilayers is the RNA. And the brighter region are those lipid-like materials. And what we found is the affinity between the RNA and the particle or in the loading material is what determines if the material is efficient as an RNA carrier or inefficient as an RNA carrier. Namely, if the RNA is not released inside the cells, the payload is inactive and then the system is inefficient. So we have to have in drug delivery or in RNA drug delivery a contradictory necessities. From one hand, you want to be able to protect the RNA en route, which can be in the blood, or in animals, it will be in the water possibly, or in their gastrointestinal tract. But then once you enter the cells, you have to release the RNA so it carries out the therapeutic activity. And we went on and tested it in animal models, and we were able to knock down different genes inside the body. This is one example that was published in Nature Nanotech. And we went on and showed it in additional cancer models. And what you see at the end of the day is this is a hepatocellular carcinoma of mice. And you can notice on the upper image a control mouse on the left-hand side and a treatment mouse on the right-hand side. And 72 hours later, the control mouse, you can actually notice the tumor continues growing, while the knockdown mouse on the right-hand side, you can notice the knockdown in this case of luciferase. So it's one gene inside a tumor. And in cancer, people have gone on and treated with multiple systems or multiple genes, and that seems very, very promising. But we were surprised because Monsanto came and picked up this technology. Monsanto is a large agricultural company, and for me, it opened my eyes saying that, wait, systems that we've developed here for medicine may be also efficient for treating other forms of disease. And do we have a common ground between treating cancer and possibly also treating hunger around the world? And if we think of it metaphorically, maybe 5% of the society are ill at one type of disease or another. However, all of us eat 3 times a day. So we do need food, and we need better and more efficient food. So the first example I'll give is how we use RNA to treat diseases in aquaculture, namely how we use RNA to treat fish and shrimp in water ponds. So shrimp disease, or approximately 20% of the global shrimp production, is inhibited today due to viral diseases. These are viral diseases that are very similar to the flu. However, the shrimp, unlike us, die once they get this disease. And one example is the WSSV, white spot syndrome. It's small dots that you can see on the shrimp itself. And for example, in the Philippines last year, they were unable to export shrimp due to this viral disease. And RNA can be an efficient mode of treating this disease. However, there is a challenge. In shrimp, unlike a salmon fish where you can inject one salmon at a time, shrimp are small, and at the end of the day, if a salmon will sell for $150, a shrimp will sell for approximately 12 cents. So there's no efficient injection system for treating shrimp. And what we had to develop is a food additive that would deliver the RNA to the shrimp in order to treat them against the viral disease. And at first, we wanted to see how long RNA can actually inhibit protein synthesis inside the shrimp. And what we found is that each treatment would be good for approximately 1 month, or a little less than 1 month. This is very, very similar of RNA knockdown we see in humans and other organisms, which lasts approximately 3 to 4 weeks. But the particle stability had to be very different. We needed to have a particle that would be stable in the ocean or in the water that we grow the shrimp. So we had to engineer a particle that would be stable in this environment. And then was the second necessity of this system. The particles had to be taken up by the shrimp gastrointestinal tract in order to induce RNA inside the body. And in a double-blind experiment we ran in the United States, you can see what really made me believe in RNA technologies a lot more than I've ever seen before. So we challenged shrimp, not in Israel. In Israel, we can't actually bring this virus in. It was done in the US. And we challenged shrimp with the virus. And as you can notice, 0%. Now, this is out of the report that this company actually produced. 0% of the shrimp survive when they have the virus. And this is what it is also in nature. However, when you treat with the RNAi treatment, you get nearly 90% survival. And that was amazing in my eyes, how you can knock down a single viral gene and reach survival in the animals. In my eyes, it gave me a lot more belief in RNA technologies for the future. However, we're still not at the same standard of injection, meaning we still have a way to go. And drug delivery in an oral manner isn't as effective as drug delivery when you inject the RNA to the animals. But that wasn't it. If we can go for shrimp, which are live, can we also go for plants? And namely, the question was, could we use nanotechnology in order to deliver or reduce the amount of pesticides we're using today for treating crops, and we all are very well aware of the harsh effects spraying has on the environment, the crops themselves, the people that eat them, but also the neighboring people that live around these fields. And we were looking at a challenge of delivering materials through the leaves down to the entire plant, because that's where the challenge actually exists. How can you go and spray a crop with less materials and still reach the same efficacy? In addition, what we learned is that many materials that are today given to crops, either through the ground or through their leaves, just don't penetrate well enough. So you need to give large amounts. And at first, we tested the ability of nanomedicines to deliver a material from the leaf to the roots. And what we're looking at here is the root of a plant 24 hours after the leaf was applied with the nanomedicine. And you see the beginning of application. So this is from the top of the plant all the way down. 48 hours later, we see more of the nanoparticles inside the roots. And 72 hours later, we have almost full coverage. Oh, yeah. And 96 hours later, we have release of the payload from the particles into the cells of the plant. So we have here a system where we can actually apply active ingredients to the leaves and treat the roots of the plant. We tested also the ability to treat from one. Yeah. We tested the ability to treat from one leaf to the others, and you can actually see here a leaf that was applied, one leaf that was applied with the nanoparticles, and adjacent leaves, the accumulation of the nanoparticles inside. So at the interest of time, I won't go into all the data. I'll just explain that what we found is that nanomedicines can actually be used for treating plants, one, from the leaf down to the roots, and two, from one leaf to the other roots. I want to thank you for your time. I will end with my thanks to, this is my team today at the, yeah, here they are. This is my team at the Technion, and thanks very much for coming here. So I'll just end with one sentence saying that I think many times we learn from humans. We have a lot of knowledge in the field of cancer research and other disease treatment using nanomedicines. Not in every time we can translate it to the clinic. However, that knowledge can be used and or applied to additional areas. And sometimes it even benefits back. Can we use a shrimp as a model for treating people? And maybe later on also, can we treat from one to another and actually benefit mankind? Thank you very much. Thanks.
For historical reasons, cancer was the first really founded project, and there is still many, many things that we learn that can be applied to other areas. Question, please.
Monsanto's Roundup.
Yeah.
Monsanto's herbicide Roundup
Yeah.
is something you spray onto the leaves, and
Right.
it actually does go down to the roots. So I'm surprised that Monsanto wouldn't already have their own vectors for doing that. But
Yes.
this is a pesticide, you're saying, rather than a herbicide.
Yeah, so actually, Monsanto's Roundup this year was pulled back from many countries because of its, it has genetic effects, and which we all assumed it may have. It's an amazing system when you wanna get rid of herbicides. However, one, we're using too much. So if we could reduce the amount by 1,000-fold, there would be a lot less impact on the environment, the water, and of course the people that are around it. And that's where we aimed. We aimed to reduce the amounts that are used today. It has a financial benefit for the agricultures. It has a financial benefit for the companies because it means a new patent. And it has a health advantage for all of us that will be consuming a lot less of these hazardous materials at the end of the day. Yeah.
Next question.
Yeah, related to that, that was a really nice talk. Financial issues. How much more expensive would you make shrimp production if you start giving them RNAi medicine?
Yeah, so I'll give the answer also for shrimp and also for crops. So in shrimp, what we can afford is 1 cent per shrimp, and we're there today. So we had to also improve our RNA production system. And don't forget, though, that unlike human medicines, there's a bit more leeway. There's a very strong regulatory process in agriculture, but there's a bit more leeway in the type of product we can actually define when we come to define a new medicine for animals. So that reduces costs, and that's in shrimp. And in our main challenge is how do we create a delivery system that will be affordable for crops. And we actually produced today a kilo of liposomes for less than $10. So, yeah. When I sell them to you, Tuan, it will cost 20. But because you can afford them, you're very well funded. But in agriculture, really, the price is what plays the role. And in addition to efficacy. So we had to go for that, and we've been working on that for the past year. How can we actually create a liposomal system that will be affordable also for agriculture? Yeah.
Coming back to the liposomal system, you showed an HTS picture of different materials that you used. And you picked out the best one, of course.
Yeah.
Can you say something about what is the pattern there? What is the mechanism? Why are some doing better than others? The second question is it the same story for shrimps, plants, etc.? I mean, is that a general picture that comes out of it, or is it dependent on the species?
Okay, yeah. So thanks for those questions. So first about the mechanism of why one material works and why another doesn't work. And actually, sometimes they're very, very similar in their molecular structure. And initially, what we thought is that some of the particles just are not taken up by the cells. However, when we ran microscopy of effective and ineffective materials, both were taken up by the cells. And what really dictates, and we know this today, very high confidence, what dictates the ability of a carrier to be effective is the affinity between the cationic backbone and the RNA. When the affinity is too high, you won't get release inside the cells. And when the affinity is medium, you'll get an effective carrier, so a small enough particle that will be stable also inside the delivery route. but you will get release inside the cell. What's medium? There are several ways of measuring it. One way is trying to introduce a negative charge, such like heparin, and running a heparin binding assay for challenging the material. Now, are we similar to plants? So I think we're closer to shrimp, but interestingly, I mean, we were shocked to see that there are a lot of similarities, at least in the efficacy of the delivery, between us and the plants. It's true that, for example, the water that the shrimp are grown in the salinity is very, very different than our blood, our circulation. So we can modify the liposomes and also decrease that way some of the price. However, some of the concepts that were developed for humans are also very applicable for other species and surely also for agriculture. So, yeah.